Hello, I'm Collin Kaufman. I'm a senior graduate student in Dr. Martha Gillette's lab, and I'm going to talk to you today showing you some of our lab space and introducing you to some of the people that work here. So, let's get started. This is our primary wet lab space. Everyone in the lab gets bench space here, where we can do everything from preparing solutions to putting the final touches on microfluidic devices. This room also includes several pieces of basic lab equipment, such as a pH meter, weigh-bench, spectrophotometer, and our very own stock of many reagents. Perhaps one of our most heavily utilized spaces is where we prepare for and perform immunohistological staining. In this room, we fix tissues with paraformaldehyde and use IHC to reveal the expression and localization of various biomolecules of interest. These biomolecules can reveal everything from gross morphological structures like the hippocampus or suprachiasmatic nucleus to specific subcellular components within individual neurons. The Gillette lab also has a strong history with electrophysiology. And through here you'll see one of the electrophysiology rigs set up inside of a Faraday cage to help isolate neuronal signals from brain structures such as the hippocampus or suprachiasmatic nucleus. This room that we're about to enter focuses on the production and imaging of various gel electrophoretic techniques such as Western blots. And here in the back we can see where some of the grad student offices are. This room is our cell culture suite where we perform animal dissections for the primary neuronal, endothelial, and muscular tissues that we study. This room is also fully equipped with a sterile biosafety cabinet, multiple incubators, and a range of other useful instruments. One of the many benefits to being in Dr. Gillette's lab is our access to lab spaces all over campus, including in this building here, the Beckman Institute. Our lab space here in Beckman is capable of performing many of the same experiments and techniques as what we have in Morrill Hall, such as electrophysiology, Western blots, and immunohistochemistry. However, this lab space also has some unique capabilities as well. For example, this tank serves as a storage unit for hundreds of millions of cells frozen in liquid nitrogen so that they can be thawed and used in experiments at a moment's notice. And next to it, in the corner, we have a cryostat, which is capable of creating brain slices as thin as 5 microns. Now that you have an idea of where we work, it's time to show you some of what we do. I'll first get started by talking about my own work studying the neuromuscular junction in vitro.
Hi, I'm Collin, and I'm a 6th-year PhD student in the neuroscience program here at the University of Illinois. My project primarily focuses on the development and characterization of an in vitro platform for the study of neuromuscular junctions. We do this through a mix of tissue engineering and classical cell culture techniques with the goal of creating a programmable biological machine. If you would like to hear more about my project, please read my publication in APL Bioengineering earlier this year. For now, I'm going to turn you over to my coworker, Miles Norsworthy, who's going to talk about his project investigating the redox state of axons and dendrites.
Hi, my name is Miles Norsworthy. I'm a graduate student in Martha Gillette's lab. My work focuses on the internal and external redox states of hippocampal neurons. With the use of redox-sensitive fluorescent proteins, I am able to measure the subcellular redox state of neurons, and with microfluidic devices, I am able to chemically isolate parts of cultured neurons to alter the redox environment. While my work uses fairly simple primary cell cultures, others in the lab use more complex three-dimensional models.
Hi everyone, I'm Amanda Weiss, a 4th-year student in the Gillette lab. I'm also co-advised by Dr. Hyunjun Kang. For my research, I've been growing spinal cord organoids, which are pictured here. These are clusters of motor neurons and glia that are grown from stem cells, and I've been evaluating their similarities and differences compared with rodent spinal cords. I'm also testing nanoparticles to see if they can alleviate stroke phenotypes. And I have a new project where I'm modeling the blood-brain barrier and helping to determine whether circadian rhythms influence susceptibility to neurological blood clots. Next, you'll hear from Harsha, who is studying a very newly discovered and important part of the body called the lymphatic system.
Hi, I'm Harsha, a senior in IB. For the past couple of years, I've been studying circadian rhythms in the lymphatic pathway. The lymphatic pathway is made up of cerebrospinal fluid, and this is used to clear up waste in between our neurons and prevent diseases like Alzheimer's from developing. Now, this pathway is heavily dependent on circadian rhythms, and so our new study is a Sweedler-Gillette collaboration where we are studying the effect of circadian rhythms on the chemical composition of cerebrospinal fluid. And so now here's Taylor to talk about astrocyte connectivity in the hippocampus.
Hi, My name is Taylor Jorgensen, and I'm a first-year neuroscience graduate student. I study how astrocytes interact with neurons in the hippocampus throughout the circadian cycle. Using the electrophysiology technique of patch clamping and injecting dye into astrocytes, our lab has found that astrocytes have a greater degree of connection to each other during the night, which is when nocturnal animals like the rats that we study are active. My goal is to determine whether disrupting this coupling between astrocytes will affect long-term potentiation response in the hippocampus. Next up, you'll hear about our work involving the blood-brain barrier from our newest lab member, Wang.
Hello everyone, my name is Wang and I'm a first year grad student from Dr. Martha Gillette lab under Molecular and Integrated Physiology department. My research is on the circadian rhythm of the blood-brain barrier in mammalian models, and I would like to learn the differences in permeability between day and night of blood-brain barrier. By understanding the blood-brain barrier oscillation, we can improve the drug delivery system for neurological diseases.
Hi, I'm Dr. Jennifer Mitchell, and I'm a senior research scientist here in Martha Gillette's lab, and I want to thank you for watching this video. If you have any additional questions, feel free to visit our website, or you can email Martha and I at the emails listed here. Thank you.